We are dividing into the fascinating world of biology, specifically DNA replication. Ever wondered how your body creates exact copies of its genetic blueprint? Well, here's the scoop. DNA replication is like the ultimate copy machine. It starts with unwinding the iconic double helix, then enzymes step in to assemble a brand new strand that perfectly complements the original. The result? Two identical DNA molecules, each ready to power the next generation of cells. It's a microscopic masterpiece of precision and teamwork, science at its finest. Now let's see how this DNA replication starts.
DNA replication is a highly coordinated process that ensures the accurate duplication of the genetic material. Here is a step-by-step breakdown of how DNA replication occurs. First is the initiation. The zoomed-in part is the origin of replication. Replication begins at specific locations on the DNA called origins of replication, where the DNA double helix is unwound. Now, the enzyme helicase unwinds the double-stranded DNA by breaking the hydrogen bonds between complementary base pairs, creating two single-stranded DNA templates. So let's see the representation on how the helicase unwinds the double-stranded DNA. We have what we call single-strand binding proteins. These proteins bind to the single-stranded DNA to prevent it from reannealing or forming secondary structures. In the RNA primer synthesis, the enzyme primase synthesizes a short RNA primer complementary to the DNA template strand, providing a starting point for DNA synthesis.
The next step is the elongation. Here, the DNA polymerase will build a new strand from the primer in a 5' to 3' direction along the helicase. Interestingly, the polymerase can only add nucleotides in the 5' to 3' direction. Do you see the strand above? That is called a leading strand. And the strand below is called a lagging strand. DNA polymerase can also add nucleotides in the 5' to 3' direction. However, it moves discontinuously, unlike in the leading strand. So, it requires multiple primers for DNA polymerase in order to form a new template strand. This will form the DNA fragments, which are also known as the Okazaki fragment.
After the elongation process, we're now moving on to the termination phase, which is the last phase of the DNA replication. So the first step here in the termination phase is the removal of the RNA primers from the two DNA strands. Once the primer was removed from the DNA strands, now comes in the DNA polymerase 1, wherein it replaces the RNA strands into a DNA strands to complete the two strands of the DNAs.
The remaining gaps between the DNA strands are then sealed with the help of an enzyme called DNA ligase. This will bind the 3' end of the nucleotide and the 5' end of another by forming phosphodiester bonds between them.
As DNA is a long molecule, some replication errors occur. But don't you worry guys, DNA has a super team to keep everything just right. Meet DNA Polymerase 1, the fixer-upper, swapping out starter pieces called primers for real DNA. Then, meet DNA ligase, the glue master. Zips everything tight so DNA is perfect. Together, they make sure our cells have the right copy of DNA instructions. And just like that, DNA replication is complete. Thanks to our amazing molecular team, our cells have a perfect copy of DNA. Hope you learned something new today. See you next time, biomeologists. Bye-bye!